Hey everyone, this is a video lecture on innate immunity. This is part of chapter 20 in our textbook and part of the lymph system chapter. So the first thing that's really important to be aware of is that there's actually two kinds of immunity. We're going to have innate immunity and we're going to have adaptive immunity. In this video lecture, we are only talking about innate immunity. And another way of describing innate immunity is by saying it's non-specific. So it's not stimulated or targeting any specific antigens. Instead, it's just going after anything that might be harmful. So let's start with some basic definitions. Immunity is the body's ability to resist and defend against infectious organisms or any other substances that could damage tissues and organs. So your immune system is also capable of working to protect you from things like toxins and poisons. It's just a case of, can it do it fast enough to keep you alive? So an immune response could be non-specific. It's not distinguishing one type of threat from another, or it could be a specific response, which is protecting you against a very particular threat. So if you think about vaccines, they are designed to give you the ability to defend yourself against a very particular pathogen. Whereas non-specific immunity that we're talking about today are the ways that your body just protects itself in general. And what we're going to find is that there's actually 7 types of or mechanisms of innate immunity in your body. So in this lecture, we're going to talk about physical barriers. We're going to talk about phagocytes. So that's number 2. The third one is immune surveillance. The fourth one is interferons. The fifth is complement. The sixth is inflammation. And the seventh is fever. So that might be a helpful way to study them in order to help you remember the different ways that your body protects you in a non-specific or non-targeted way. So first practice question, defense of the body against a particular pathogen is provided by. So that would be D, specific immunity, because it's referencing a particular pathogen. And again, in this lecture, we're going to focus on the innate immune system or non-specific immunity. So let's start with that first mechanism, which are physical barriers. And this is going to include the skin and mucous membranes, often referred to as the first line of defense, because the integumentary system is a major barrier for potential pathogens to breach. You have a number of ways that your skin protects you from potential pathogens. One is the secretions from your sweat glands and your sebaceous glands that you learned about way back when you did the integumentary system in A&P1. They have secretions, and those secretions can help wash away microorganisms, but they can also contain things like lysozymes and bactericides, things that are actually harmful to pathogens. Your hair can help protect you from abrasion and even prevent bugs from reaching your skin and biting you. And the actual keratinocytes of the stratified squamous epithelium of the skin, those cells are linked together with desmosomes. And so that really helps lock out potential invaders. Inside your mouth, vaginal canal, anal canal, things like this, the urinary system, we're going to have some mucous membranes, and that mucus is going to play a really important role because it's very sticky and it has a low pH, so it's acidic. It may also contain enzymes and antibodies, and the cells themselves have a lot of tight junctions, so all of those characteristics are going to help protect you from potential pathogens. Now, The second mechanism are cells that can engulf pathogens or other debris that they might come across. And there's three kinds of cells we want to know about when we think about phagocytes. We want to know about macrophages, eosinophils, and neutrophils. So all three of them are capable of phagocytic behavior. That is, they can engulf through endocytosis another cell or debris, but they have different origins. So eosinophils and neutrophils are leukocytes or white blood cells. Macrophages are going to come from monocytes, so a little bit different origin there. However, they do share some characteristics. All phagocytes are capable of emigration or diapedesis. And you hopefully remember that term. It means they can exit the capillaries by squeezing between the endothelial cells. They exhibit chemotaxis, so they can either respond by moving towards or away from particular chemical signals. They are all capable of adhesion. So they will attach to their target using their receptors to bind to the surface of that target. But that the way they bind to things is a bit arbitrary. So again, they're not targeting a specific pathogen. They are just targeting any pathogen. And they have two things that they might do once they attach to it. Some phagocytes will actually destroy the target, whatever it is that they've adhered to, but some of it will actually just flag it for other phagocytic cells to destroy. So, different things, different jobs for different cells. So you can see this little chart includes these three types of phagocytes. You can see that monocytes actually exist as two kinds. Some are fixed. If they are fixed macrophages, that means they live in a particular tissue and they stay there, whereas free macrophages are capable of meandering the body on the search for things to target and destroy. Of course, neutrophils are quite abundant, and they will go after cellular debris or invading pathogens. Eosinophils are a little bit different. There's not as many of them, and also they tend to phagocytose pathogens that are already covered in antibodies. Now, they are not recognizing any particular antibody, just antibodies in general attract eosinophils to destroy or phagocytose that cell. Our third mechanism is immune surveillance by natural killer cells. So natural killer cells are always on the hunt for abnormal cells in your body. Those abnormal cells may be your own cells that have been infected with a virus, or they could be your own cells that have become cancerous. But what natural killer cells should not pay attention to are any of the body's own cells. So they have the ability to recognize self, is how we describe that. But when they spot cells that are abnormal, either because they are infected with a virus or they have become cancerous, then they will target those cells for destruction. Now, they do have a way of recognizing abnormal cells because typically speaking, cells that are infected with viruses will display antigens that tell the NK cells they've been infected. Again, it doesn't say what they've been infected with, just that they've been infected by a virus. Or in the case of cancer cells, they will often display tumor-specific antigens on their cell membrane that the NK cells will recognize. And that's really important because in the case of cancer cells, some of the worst cancers that are the most difficult to treat are the ones who don't cause a tumor-specific antigen to be displayed. So that makes it hard for your NK cells to see them and destroy them, but it also means we don't have a target for medicines to focus on. So it's kind of a double whammy. Now, I do just want to share, I'm going to see if I can blow this image up a little bit. How NK cells actually destroy abnormal cells is, I think, kind of cool. Bear with me. All right, so here is our natural killer cell, and here is an abnormal cell that is displaying some antigens on its surface, and that's what's activating or allowing the NK cell to recognize that this other cell is not normal. So what the NK cell does, check this out. Do you remember learning about the cell organelles in AMP1? And one of those things was the Golgi body. So the Golgi body actually reorients itself with its trans face adjacent to the target cell. And then the NK cell adheres to that target cell. So now they're really close together. And the Golgi is going to create a whole bunch of vesicles filled with something called perforin. So these are proteins that are coded for by a gene in your DNA, which can, an mRNA copy can be made in the nucleus, and then it will exit out the nuclear pore. It'll go to a ribosome. We will translate that mRNA. It will then be inserted into the rough ER, where it will get folded and maybe modified a little bit there, and then it will travel via a vesicle from the rough endoplasmic reticulum to the Golgi, and then the Golgi is going to package up all these perforins into little vesicles, and it's going to wait until that NK cell adheres to a target cell, and then the Golgi will orient itself to be right next to that target cell, and then it will release all these vesicles filled with perforins, and the perforins will diffuse across that very tiny space between the two cells and literally poke holes in the abnormal cell. It'll perforate the cell membrane of the target cell, and all of the stuff inside the target cell starts to leak out of the cell, and so it ends up lysing the cell, and the target cell falls apart and it's destroyed. So you can see here is a little blow up of the vesicles coming from the Golgi filled with perforin being exocytosed into the little space, and then they start to create these little pores on the abnormal cell that ultimately will result in that cell being destroyed. All right, I just think it's a cool little mechanism. So what is perforin? So hopefully you might have picked B. It is a protein produced by natural killer cells. That is a true statement. It doesn't really say a whole lot about what it does. So this is a great example of a question that's like indicating to you that, apparently it's important to know that NK cells produce perforin, and probably that means I need to know a little bit about perforin too. All right, so our next mechanism, I got a little ahead of myself. The next thing I want to talk about is how might cancer cells evade natural killer cells? So I kind of hinted at it earlier, but there's a couple of ways that cancer cells can evade natural killer cells. So one way is that sometimes cancer cells form a tumor that has a capsule around it, and that capsule kind of protects it from the NK cells. So the NK cells are just literally not able to see the cancer cells because the capsule, which is usually connective tissue, prevents them from kind of getting access to those cells. But the definition of cancer is that cancer cells will leave the tumor and start to spread, and that's what a malignant cancer is. So once they leave the tumor, then the NK cells should be able to detect them and destroy them, which would, in theory, prevent any additional tumors from forming. But sometimes what can happen is that when the cancer cells are undergoing mitosis, which they're doing a lot because that's what cancer is, uncontrolled cell growth, sometimes during one of those mitotic divisions, a mutation occurs that results in a daughter cell that doesn't display a tumor-specific antigen. And if that happens, that means that the NK cells can't see it. That cell is essentially invisible. It just looks like a normal cell. And now that daughter cell is going to continue undergoing mitosis and creating additional daughter cells, all of whom are essentially wearing this cloak, making them invisible to NK cells. And so they're going to be able to spread throughout the body and establish secondary tumors. And that's problematic in a number of ways. Okay, our next mechanism is interferons. Now, these are chemicals or proteins that coordinate defense against viral infections. We're going to see that there's 3 different kinds of interferons. We're going to have an alpha, a beta, and a gamma. So we'll come back to that in a second. But basically, these are proteins that are released by activated lymphocytes, macrophages, or infected cells themselves. So there are several sources of interferons. What they do is bind to receptors on normal cells, and they will trigger an antiviral protein production inside the cell. And they will stimulate macrophages and NK cells to go, to be like super active and on the hunt for infected cells. So this is important for a few reasons. Number one, if one cell is infected with a virus, then we want to let macrophages and NK cells know that, right? We want them being on the lookout for abnormal cells that are infected with a virus. But we also want to try to protect our friend cells that live nearby. So if I'm a cell in the nose and I just picked up a cold virus, I want to try to protect my friends, my nearby cells. So if I release interferons because I've been infected with a virus, the interferon will go out and bind to receptors on the other cells right around me and say to them, hey, there's a virus in the neighborhood. You need to protect yourself. So you need to start producing proteins that will interfere with viral replication if you get infected by a virus. So unfortunately, interferons don't prevent infection from the virus, but they do make it harder for the virus to replicate inside any cell that has received the interferon message. So, it's a good thing for sure. Now, another way that we describe interferons are that they are cytokines. And cytokines is just another vocabulary word, and it just means any chemical that's involved in local cell signaling. So it's a way that cells living in the same basic neighborhood can communicate with one another about things that are going on locally. Now, in terms of those three different types, the alpha interferon is produced by cells that have been infected with viruses, and that will attract and stimulate NK cells and enhance viral resistance. The interferon beta is going to be secreted by fibroblasts, and that's going to be important because beta will actually help slow inflammation in a damaged area. Inflammation is a tricky thing, and we're going to talk about it during this lecture, but inflammation is both kind of good and then also bad, so you want a way to shut it down. So interferon beta is going to be our mechanism for that. Remember, fibroblasts are the cells found in connective tissue. And then the last type is interferon gamma. That's going to be secreted by T cells, and we'll learn more about T cells when we talk about adaptive immunity. It's also going to be released by NK cells, and in both cases, that gamma will activate and stimulate the activity of macrophages. So again, think about the big description here. Interferons are chemicals that coordinate defense against viral infection. So they work in a few different ways, but the goal for all of that is to help prevent and reduce viral infection. Now, another kind of protein, another group of proteins is something called complement, which is a very odd name. Sometimes it's called the complement system, if that makes more sense. These are a whole bunch of proteins, and they are going to assist or complement antibodies. Now, antibodies have to do with adaptive immunity. So complement itself is not specific, but it does support antibody-mediated immunity, which is specific. So there's a little bit of a crossover here. And complement is going to act in cascades. If you recall the clotting cascades, it's going to be a little bit similar to that. So we're going to have three cascades or three pathways by which complement can be activated. We're going to have the classical pathway, we're going to have the lectin pathway, and we're going to have the alternative pathway. And I just want to tell you right now that all three pathways are going to result in splitting inactive C3 proteins to form C3b and C3a proteins that are activated. So I'm going to say that one more time. All three pathways will lead to the creation and activation of C3b and C3a. The details of each pathway, I'll be honest, not that important. But the end result is because C3b, once activated, will bind to bacterial surfaces and enhance phagocytosis. So notice I just said bacteria, and when we were talking about interferons, it's all about viruses. So that's a big difference between interferon and complement. Interferons, think viruses. Complement, think bacteria. All three pathways will lead to the activation of C3b, and C3b will bind to bacteria cell membranes and then enhance phagocytosis by phagocytic cells like macrophages, eosinophils, and neutrophils. The other thing that happens is C3a also gets activated, and C3a is really important in the inflammation response. So we are going to see that C3a will activate inflammation. C3b will activate the destruction of bacterial cells. And I think if you know that much, I think you'll be good. You are welcome to study these diagrams and learn a little bit more about how we can take C3 and split it into C3a and C3b. But again, in the end, all three pathways activate C3b and C3a. All right. The process by which the surface of a microorganism is covered with antibodies and complement, rendering it more likely to be phagocytosed, is called. And the word that I didn't mention on the last slide is opsonization. And this is a very important vocabulary word. It's a strange word, opsonization. But that is what we say when the complement system binds to a bacteria cell that already has antibodies on it, making it more likely to be phagocytosed by a neutrophil, by a macrophage, by an eosinophil. That is called opsonization. All right, so now we are on the sixth mechanism, I think, which is inflammation that I've mentioned. So this inflammation process may have been initiated by C3a because C3a activates the inflammatory response. It does that because it activates mast cells and basophil cells to release histamine. So our mast cells will release histamine and our basophils will increase the degree. Let me try that sentence again. The C3a will activate mast cells and basophils to release histamine, which will in turn result in inflammation in that area. In turn, that inflammation, and this goes all the way back to AMP1, when we first learned about inflammation, will attract and activate phagocytes. It's going to activate or stimulate blood flow to the region because we're going to get the dilation of blood vessels as a result of the histamine and also things like heparin that'll be released. If there's been any kind of injury that comes along with this pathogenic invasion, we may need to form a clot, if you remember that from AMP1. And of course, we want lots of those phagocytes both to go after any pathogens, but also to clean up any cellular debris from any injury in the site. So we've got a nice little flow chart here showing us we've got this tissue damage that may have introduced pathogens. So that's going to result in a chemical change in the interstitial fluid, which is going to release histamine and heparin from mast cells and basophils. We've also got activation coming from C3a. So again, we're kind of getting like a double activation here. And again, that's going to result in phagocytic attraction and then also redness, swelling, heat, and pain. And ultimately what we hope to see is repair of the tissue. Things to note that when you have a potential pathogenic invasion or an injury to the tissue like this, any of the cells that are damaged are going to release things like prostaglandins, proteins, and potassium, and that's what leads to the change in the interstitial fluid, which in turn can activate those mast cells. At the same time, they are also going to be receiving messages from the complement system if it's been activated, and then that C3a will also activate those mast cells. So, notice in the body, there's almost always a backup system. There's very rarely only one way to achieve a particular outcome. All right, and that brings us to our last mechanism, which is fever. You're probably all familiar with that. I'm sure most of you, if not all of you, have had at least one in your lifetime. So this just is an elevation of your normal body temperature as a result of increased metabolism. That is a fancy way of saying increased chemical reactions inside your cells. We typically consider fever anything that's greater than 99 degrees. And when someone has a fever, we find pyrogens circulating in their body, and those are fever inducing proteins. They actually affect the thermostat in the hypothalamus. The hypothalamus, of course, is a major center in the brain, and one of the things it does, among many, is help regulate body temperature. So the pyrogens kind of reset that thermostat so that your body can get hot. This is great because it's going to increase the metabolic rate, which means your cells are going to be more active, which may help your immune cells to be more active in going after whatever is infecting you. And it also may actually inhibit some viruses and bacteria because that a fever temperature for us might actually be too high for their proteins, and it may lead to the denaturation of some of their enzymes. On the other hand, if our fever gets too high, we could start denaturing our own enzymes, and that's not a good thing. So on the one hand, we want enough of a fever that our tissue defenses can be mobilized more rapidly and the repair process can be sped up. But what we don't want is for it to be so high that we actually damage our own cells. So it's a fine line there. All right, a sample of John's blood shows a high level of pyrogens. This would indicate that John hopefully you said C is running a fever. By the way, hopefully you all recognize the word pyrogen, like a pyromaniac or pyrotechnics have to do with fire. So, or, heat. So that would be a fever. All right, and then I've included this last slide. It's kind of running off it's running off our slide there a little bit of our mechanisms. So we have that physical barrier. Let's see if I can blow this up a little bit. The answer is no, I cannot. All right, so we've got physical barriers, phagocytes, immune surveillance, interferons, complement system, inflammation, and fever. And all of these are non-specific defenses. They're just trying to protect you from everything kind of equally, if you will. They're not targeting any specific pathogen, and so therefore we call them non-specific immunity. So the body's non-specific defenses include all of the following except and hopefully you picked B and T cells. They are going to be really important in specific immunity or adaptive immunity, and we're going to talk about that in the future. So I hope this presentation helped you master this content, and I will be available in office hours for questions.